speaker again, and we're going to move to the next talk. The title of the next talk, and which is the final talk of this morning, is Sex Differences in the Human Connectome. Title is fairly long, so I have to truncate it. And the talk is going to be given by Dr. Prasad.
Thanks for the introduction, Joe. The paper I'm describing today uses a novel tractography algorithm on 234 young adult twin subjects to understand sex differences in the human connectome. This is work done by Neda Jahanshad and our colleagues at the Laboratory of Neuroimaging, UCLA. We can understand the structural connectivity of the brain using standard magnetic resonance imaging and diffusion weighted imaging. And we can use a tractography algorithm to extract fibers from our diffusion-weighted images and use those fibers to understand connectivity within and across hemispheres. There's literature that shows diseases like Alzheimer's and schizophrenia change the structural connectivity of the brain significantly. Our subjects consisted of 234 young adult right-handed twins and their siblings. We imaged them using 4 Tesla standard T1 weighted magnetic resonance imaging and high angular resolution diffusion imaging, referred to as HARDI. The T1 images were corrected for non-uniformity, and we used eddy and epi correction on our diffusion weighted images. And then linearly registered the HARDI images to the T1 image space. For each of our T1 images, we extracted a cortical surface, parcellated it automatically using FreeSurfer to find 35 different regions in each hemisphere, and then downsampled these parcellations into the diffusion-weighted image space, and then dilated them. This is to ensure that fibers we calculate from tractography will intersect these parcellations. And then we calculated fibers in each of our HARDI images using a novel tractography algorithm, which resulted in 2,000 to 5,000 fibers per subject. This slide gives an overview of our pipeline. We linearly align our diffusion-weighted images to the T1s, calculate fibers, and then we parcellate the cortex of our corresponding T1 images, dilate them, and then find the intersection of the fibers with the different regions in the cortex. And we use this information to create a connectivity matrix. The single tensor model of diffusion imaging doesn't account for fiber mixing or crossing. The information that we gain using HARDI images creates an orientation distribution function, or ODF, at each voxel. And using that information, we can delineate multiple fibers that go through a voxel that differ in their orientation. And there are some estimates that 40% of the voxels that we get from diffusion images have fiber crossing. So it is useful and it's advantageous to have this higher resolution diffusion imaging. Our novel HARDI tractography method scores curves in our diffusion-weighted image space using the orientation distribution functions and the prior probability of the seed points used to generate the curves. Our ODFs are generated using a normalized and dimensionless estimator derived from cue ball imaging, and our curves are seeded randomly throughout the diffusion-weighted image space using a prior probability based on the fractional anisotropy in the image. Our method exhaustively searches curves throughout the image space at a particular resolution. We use the voting process provided by the Hoff transform to decide the best scoring curve out of that set. And we keep these curves as our final result from the tractography method. This matrix shows the connectivity results from the fibers we calculated from tractography. And the axes show the 35 different regions we parcellated in each hemisphere. So there's 70 on each side. It's a symmetric matrix, and the diagonals represent the connectivity within regions of the cortex, while the other parts of the matrix show connectivity between two regions. This can be between the left and right hemisphere or within a hemisphere. The color bar shows a measure of the connectivity, with the blue area showing a proportion of the fibers that, a small proportion of the fibers that connect to a region, while the red area show larger proportions. We thresholded our results by removing any connectivity measures where they weren't shown in 95% of our subjects. And we averaged the proportion of overall fibers across all of our subjects to get this matrix. The upper left square block and the bottom right block show the connectivity within each hemisphere, the left and right. And then the bottom left and top right show the connectivity across hemispheres. Our results show that most of the connectivity is within hemispheres, and this agrees to literature that analyzes connectivity in the brain. We looked at a sub-matrix to analyze sex differences. In our case, we focused on the frontal cortex. And the frontal lobe is important in mediating executive function, self-control, and speech. And in our extracted frontal cortex, we selected 5 different regions to understand sex differences in connectivity. Literature shows that brain structure does differ significantly across sex, and that linguistic processing and cognitive traits have neuroanatomical correlates depending on sex. There's also evidence for sex-dependent functioning. We use the fibers from tractography to analyze the connectivity dependent on sex in the frontal cortex. The figure on the right shows the p-values of the significance, the sex differences in connectivity across the frontal cortex, between and within hemispheres. We use a random effects regression to account for the interrelatedness of families in our subject set. Our results show that the highest connectivity was found in the superior frontal cortex, or the highest significance in sex difference was found in the superior frontal cortex, with women having higher connectivity, while men had higher connectivity in the the right lateral orbitofrontal cortex and the connection between that region and the ipsilateral insula. Literature shows that genes are responsible for a significant variability in the cortical structure and the white matter fiber microstructure. We leveraged the monozygotic and dizygotic twins in our subject data set to understand heritability through our connectivity matrices. And then we validate whether the connectivity matrices provide an effective tool for future genetic studies of cognition and disease. Monozygotic twins share 100% of their genes, while dizygotic twins share 50% of their genes on average. We use Falconer's heritability to gain a rough initial measure of heritability in our subjects. It looks at the interclass correlation between the monozygotic twins versus the interclass correlation in the dizygotic twins. The figure on the right shows the false discovery rate adjusted p-values of our monozygotic twins in blue and dizygotic twins in green. It shows that the monozygotic twins have higher connectivity and more significant p-values, and that there's a significant difference between the monozygotic set and dizygotic set, showing that our connectivity matrix is able to extract heritability information from our subjects. This figure goes into detail of the heritability in our 35 different regions that we parcellated in the cortex. The color bar shows our Falconer's heritability percentage, so the blue means that it's not very heritable and the red means it's more heritable. And we use the same sex subjects in our dizygotic twins to remove any gender bias. Our results show that the highest heritability was found in the left precuneus, with the left pericalcarine fusiform gyri and left cuneus having moderate heritability. To summarize the results from our method, we used a novel Hardy tractography algorithm and applied it to 234 healthy young twins and their siblings. We used the fibers from the method to analyze sex differences in the structural connectivity in the frontal lobe, and our results agreed with literature studying the connectivity of the brain. We also established the viability of a connectivity matrix in extracting heritability information from subjects where we have monozygotic and dizygotic twins, and its future use in genetic studies relating to cognition and disease. Our future work will look at general and global conclusions from our connectivity matrix, and we'd like to understand how our results are affected if we use a different type of tractography method. For example, we could use streamlines instead of our Hardy method. We'd also like to understand the subcortical connectivity dependent on these fibers we calculated, in addition to the connectivity of regions in the cortex that we focused on in this project. And we'd like to incorporate structural equations into our genetic analysis to make our estimates more precise. At the Laboratory of Neuroimaging, we also have an Enigma project which incorporates tens of thousands of subjects, which can improve our genetic analysis. I thank you for your attention, and please ask any questions you have.
Thank you very much. Now the paper is open for discussion. Actually, a quick question. And when you are comparing the sex and differences, have you matched the age and IQ levels between male and female groups?
You mean account for that variability? No, we didn't change. We didn't account for that, but we did. Let's see. I think we did look at a subset of our initial population of subjects and tried to normalize it so that they were within the same age range.
More questions? Let me just ask one more question. For the Hardy acquisition, and typically the data acquisition time is fairly long.
I think it's 24 minutes per subject.
Okay. So do you see in the future, and this particular technique can be widely used in normal clinical setting?
I suppose it would just. I mean, I guess a doctor would be better at answering that. But I mean, if we can apply correction to the images that fix any movement happening and make those algorithms more effective, then it shouldn't be a problem.
More questions? And if not, I'd like to thank all the speakers in this morning session, and one more time. And we started in this room by just 5 or 6 people. So at the end of the session, we have lots of more people, and which is a really good sign. And thank you very much for coming, and enjoy the nice weather in Chicago.